The potato is a root vegetable native to the Americas, a starchy tuber of the plant Solanum tuberosum, and the plant itself is a perennial in the nightshade family, Solanacea. Wild potato species, originating in modern-day Peru, can be found throughout the Americas, from Canada to southern Chile. The potato was originally believed to have been domesticated by indigenous peoples of the Americas independently in multiple locations, but later genetic testing of the wide variety of cultivars in wild species traced a single origin for potatoes. In the area of present-day southern Peru and extreme northwestern Bolivia, from a species in the Solanum brachicole complex, potatoes were domesticated approximately 7,000,000 years ago in the Andes region of South America, where the species is indigenous. Some close relatives of the potato are cultivated. Potatoes were introduced to Europe from the Americas in the second half of the 16th century by the Spanish. Today they are a staple food in many parts of the world and an integral part of much of the world's food supply. As of 2014, potatoes were the world's fourth largest food crop after maize, corn, wheat, and rice. Following millennia of selective breeding, there are now over 5,000 different types of potatoes. Over 99% of presently cultivated potatoes worldwide descended from varieties that originated in the lowlands of south-central Chile. The importance of the potato as a food source and culinary ingredient varies by region and is still changing. It remains not an essential crop in Europe, especially Northern and Eastern Europe, where per capita production is still the highest in the world, while the most rapid expansion in production over the past few decades has occurred in Southern and Eastern Asia, with China and India leading the world in overall production as of 2018. Like the tomato, the potato is a nightshade in the genus Solanum, and the vegetative and fruiting parts of the potato contain the toxin solan, which is dangerous for human consumption. Normal potato tubers that have been grown and stored properly produce glycoalkaloids in amounts small enough to be negligible to human health, but if green sections of the plant, namely sprouts and skins, are exposed to light, the tuber can accumulate to high enough concentration of glycoalkaloids to affect human health. Potato plants are herbaceous perennials that grow about 60 centimeters, 24 in high, depending on variety, with the leaves dying back after flowering, fruiting and tuber formation. They bear white, pink, red, blue, or purple flowers with yellow stamens. In general, the tubers of varieties with white flowers have white skins, while those of varieties with color flowers tend to have pinkish skins. Potatoes are mostly cross-pollinated by insects such as bumblebees, which carry pollen from other potato plants, though a substantial amount of self-fertilizing occurs as well. Tubers form in response to decreasing day length, although this tendency has been minimized in commercial varieties. After flowering, potato plants produce small green fruits that resemble green cherry tomatoes, each containing about 300 seeds. Like all parts of the plant except the tubers, the fruit contain the toxic alkaloid solan and are therefore unsuitable for consumption. All new potato varieties are grown from seeds, also called true potato seed, TPS, or botanical seed, to distinguish it from seed tubers. New varieties grown from seed can be propagated vegetatively by planting tubers, pieces of tubers cut to include at least 1 or 2 eyes, or cuttings, a practice used in greenhouses for the production of healthy seed tubers. Plants propagated from tubers are clones of the parent, whereas those propagated from seed produce a range of different varieties. There are about 5,000 potato varieties worldwide. 3,000 of them are found in the Andes alone, mainly in Peru, Bolivia, Ecuador, Chile, and Colombia. They belong to 8 or 9 species, depending on the taxonomic school. Apart from the 5,000 cultivated varieties, there are about 200 wild species and subspecies, many of which can be crossbred with cultivated varieties. Crossbreeding has been done repeatedly to transfer resistances to certain pests and diseases from the gene pool of wild species to the gene pool of cultivated potato species. Genetically modified varieties have met public resistance in the United States and in the European Union. The major species grown worldwide is Solanum tuberosum, a tetraploid with 48 chromosomes, and modern varieties of the species are the most widely cultivated. There are also 4 diploid species with 24 chromosomes: S. stanatimum, S. forja, S. goniacalix, and S. egenderi. There are 2 triploid species with 36 chromosomes: S. chawa and S. tsuzuki. There is 1 pentaploid cultivated species with 60 chromosomes: S. cratylum. There are 2 major subspecies of Solanum tuberosum: Andigena, or Andean, and tuberosum, or Chilean. 23. The Andean potato is adapted to the short day conditions prevalent in the mountainous equatorial and tropical regions where it originated. The Chilean potato, however, native to the Quilla archipelago, is adapted to the long day conditions prevalent in the higher latitude region of southern Chile. 24. The International Potato Center, based in Lima, Peru, holds not a so-accredited collection of potato germplasm. The International Potato Genome Sequencing Consortium announced in 2009 that they had achieved a draft sequence of the potato genome. The potato genome contains 12 chromosomes and 860 million base pairs, making it a medium-sized plant genome. More than 99% of all current varieties of potatoes currently grown are direct descendants of a subspecies that once grew in the lowlands of south-central Chile. Nonetheless, genetic testing of the wide variety of cultivars in wild species affirms that all potato subspecies derive from a single origin in the area of present-day southern Peru and extreme northwestern Bolivia, from a species in the Solanum brachicole complex. The Wild Crop Wild Relatives Breeding Project encourages the use of wild relatives in breeding programs. Enriching and preserving the gene bank collection to make potatoes adaptive to diverse environmental conditions is seen as a pressing issue due to climate change. Most modern potatoes, grown in North America, arrived through European settlement and not independently from the South American sources, although at least one wild potato species, Solanum fendleri, naturally ranges from Peru into Texas, where it is used in breeding for resistance to an nematode species that attacks cultivated potatoes. A secondary center of genetic variability of the potato is Mexico, where important wild species that have been used extensively in modern breeding are found, such as the hexaploid Solanum demicin, as a source of resistance to the devastating late blight disease. Another relative native to this region, Solanum bulbocostinum, has been used to genetically engineer the potato to resist potato blight. Potatoes yield abundantly with little effort, and adapt readily to diverse climates as long as the climate is cool and moist enough for the plants to gather sufficient water from the soil to form the starchy tubers. Potatoes do not keep very well in storage and are vulnerable to molds that feed on the stored tubers and quickly turn them rotten, whereas crops such as grain can be stored for several years with a low risk of rot. The food energy yield of potatoes, about 95 gigajoules per hectare, 9.2 million kilocalories per acre, is higher than that of maize, 78 GJ/ha or 7.5106 kcal/acre, rice, 77 GJ/ha or 7.4106 kcal/acre, wheat, 31 GJ/ha or 3106 kcal/acre, or soybeans, 29 GJ/ha or 2.8106 kcal/acre. There are close to 4,000 varieties of potato including common commercial varieties, each of which has specific agricultural or culinary attributes. Around 80 varieties are commercially available in the UK. In general, varieties are categorized into a few main groups based on common characteristics, such as russet potatoes, rough brown skin, red potatoes, white potatoes, yellow potatoes, also called Yukon potatoes, and purple potatoes. For culinary purposes, varieties are often differentiated by their waxence. Flory or mealy baking potatoes have more starch, 20,122%, than waxy boiling potatoes, 16,118%. The distinction may also arise from variation in the comparative ratio of two different potato starch compounds, amylose and amylopectin. Amylose, a long-chain molecule, diffuses from the starch granule when cooked in water, and lends itself to dishes where the potato was mashed. Varieties that contain a slightly higher amylopectin content, which is a highly branched molecule, help the potato retain its shape after being boiled in water. Potatoes that are good for making potato chips or potato crisps are sometimes called shipping potatoes, which means they meet the basic requirements of semitivoridal characteristics, being firm, fairly clean, and fairly well-shaped. The European Cultivated Potato Database, ECT, is an online collaborated database of potato variety descriptions that is updated and maintained by the Scottish Agricultural Science Agency within the framework of the European Cooperative Programme for Crop Genetic Resources Networks, ECP/GER, which is run by the International Plant Genetic Resources Institute, IPRI. 37 dozens of potato cultivars have been selectively bred specifically for their skin or, more commonly, flesh color, including gold, red, and for varieties that contain varying amounts of phytochemicals, including carotenoids for gold/yellow or polyphenols for red or blue cultivars. Carotenoid compounds include provitamin A, alpha-carotene and beta-carotene, which are converted to the essential nutrient, vitamin A, during digestion. Anthocyanins mainly responsible for red or blue pigmentation in potato cultivars do not have nutritional significance, but are used for visual variety and consumer appeal. In 2010, potatoes were bioengineered specifically for these pigmentation traits. Genetic research has produced several genetically modified varieties. New Leaf, owned by Monsanto Company, incorporates genes from Bacillus thuringiensis, which confers resistance to the Colorado potato beetle. New Leaf Plus and New Leaf E, approved by U.S. regulatory agencies during the 1990s, also include resistance to viruses. McDonald's, Burger King, Frito-Lay, and Procter & Gamble announced they would not use genetically modified potatoes, and Monsanto published its intent to discontinue the line in March 2001. Waxy potato varieties produce two main kinds of potato starch, amylose and amylopectin, the latter of which is most industrially useful. BASF developed the Amflora potato, which was modified to express antisense RNA to inactivate the gene for granule-bound starch synthesis, not enzyme which catalyzes the formation of amylose. Amflora potatoes therefore produce starch consisting almost entirely of amylopectin, and are thus more useful for the starch industry. In 2010, the European Commission cleared the way for Amflora to be grown in the European Union for industrial purposes only, not for food. Nevertheless, under EU rules, individual countries have the right to decide whether they will allow this potato to be grown on their territory. Commercial planting of Amflora was expected in the Czech Republic and Germany in the spring of 2010, and Sweden and the Netherlands in subsequent years. Another GM potato variety developed by BASF was Fortuna, which was made resistant to lake blight by adding two resistance genes, BLB1 and BLB2, which originate from the Mexican wild potato Solanum bulbocostumum. In October 2011, BASF requested cultivation and marketing approval as a feed and food from the EFSA. In 2012, GMO development in Europe was stopped by BASF. In November 2014, the EFSA approved a genetically modified potato developed by J.R. Simplot Company, which contains genetic modifications that prevent bruising and produce less acrylamide-1 than conventional potatoes. The modifications do not cause new proteins to be made, but rather prevent proteins from being made via RNA interference.